It's my real pleasure to have one of my mentors here. John and I first met in 1992, I think, at least as best we can recall. And John is one of those people who is eclectic in background, but very knowledgeable about a huge number of fields. And he is, his insight was such that it led to one of the major projects in biology education. It was and is called the BioQuest Project. And it came not just with an emphasis on how one incorporates quantitative ideas in biology education, but with a whole pedagogy about how to do that appropriately. And I don't know if he's going to say anything about that, but no, he's not. But you should ask me or ask him if you'd like. John has spent a large fraction of his career on the faculty at Beloit College, where he headquartered the BioQuest project for a long time. But he did one of those things that I hope to do at some point, which is to fail at retirement, and moved after leaving Beloit to basically be the first director of a major interdisciplinary center and building at the University of Delaware, and he's been there for about 3 years. Is that right? 7. Oh God! See, 7 years, and I can't even. can't keep track. And I've warned him. I'm going to ask him to do something. So in 1994, John was here for a workshop, and it was a workshop on biology and quantitative thinking. And at the end of that workshop, he came up to the front and he said, "Lou, I really want to thank you for what you've done." And he gave me this. And so I've had it for like 25 years. And I think I've had it long enough and learned about as much from it in my limited way as I can. So I thought it would be appropriate for John to pass this on to Shelby Scott as a new generation, and he can display for you what it does. Okay. Come on up, Shelby.
Okay, Shelby.
Okay.
This is
So, I'm going to give 2 talks today, and it shouldn't scare you that usually I try to do about 350 slides in 25 minutes. I at AAAS once did 270 in 20 minutes, but I'm going to try and be good to you today, kind of thing. Do about 2 20-minute talks so we have some questions, and don't worry, that usually means I have to skip a few here and there, but still try to make a couple of points. So you have your intellectual research interests, but obviously talked about education. And I'm an old basketball player, and one of the things I learned spatial reasoning is I would blindfold and dribble up and down the court between my legs and behind my back. That I'm only 5'17", kind of thing, that I was just a small guard kind of thing. And that being slow but being strong, I had to learn to play a different kind of way. But that kind of tactile learning and sleeping with a basketball as a teenager, kind of thing, is that spatial, three-dimensional kind of reasoning has always been a huge part of mine. And so Lou's former student, Holly Gaff, and I have written about mathematical manipulatives for learning and so on. But combining my research and teaching interests together has always been one of my aspirations. But this is an old story. And obviously, Lou, his wife being a poet, and my love of poetry, I go back to a poet, Wallace Stevens, connoisseur of chaos. A violent order is disorder, and a great disorder is an order. These two things are one. And I always wondered, boy, did he understand thermodynamics at that level, kind of thing. It's pretty impressive. But in 1944, a famous book was written by Aaron Schrodinger, it's the middle of World War II, but he's in England, and here's the Schrodinger wave equation, and he presents this little book, What is Life? And after World War II, many physicists said this was the book that motivated them to move from physics to biology, that they're going to maybe search for new laws. And he invented this notion of negentropy. Now, many people feel that he, led to Watson and Crick and the gene and, the aperiodic crystal and so on. But this notion of negentropy, I think, just did a great deal of damage in biology. And too many of our biology educators, you always got to do work. You got to spend ATP or GTP in order to do anything in biology. And it's one of the most misleading notions ever for me in terms of understanding biology. And you'll see things like this, that life is driven by the subconscious desire to fight against entropy, and, that life, matters because it's fighting against entropy. And there could not be a notion that's more wrong in biology. And so in almost 50 years ago, I wrote this article, Thermodynamics of Self-Assembly, an empirical example relating entropy and evolution. And I just was struggling with it, that why is it that people had such a misconception? And later in '75, one of my heroes, Max Loeffler, wrote a book called Entropy-Driven Processes in Biology, and here's a more recent article in terms of that Max was actually able to do burst bomb calorimetry in terms of measuring the kind of entropic changes that occur and so on. And now there's even better kind of techniques for doing this at a micro scale. But self-assembly was not much of the literature at that time. So Govind Menon at Brown in Applied Math has had a huge impact on me. But he did this nice analysis that you can look at the time that I published in early 1970s, that at the very beginning of this kind of curve and how it skyrocketed. Think about not only in terms of self-assembly, but Belousov-Zhabotinsky reactions, Turing's, Gear-Meinhardt, etc. The notions of having designs without designers has become a major part of mathematical biology. And that it's very difficult for people to kind of understand, how does disorder in nature drive order in biology? And so I've struggled to make this kind of thing much more obvious. And so for most cases, it's a simple hydrophobic effect, that you have these protein molecules with water tightly clinging to them, and that if the proteins associate with the proteins, usually by weak kind of forces, van der Waals forces, salt bridges, hydrogen bonds, etc. But the release of the water, bound water, to have translation and rotational kind of freedom is that you get organized kind of structures. And this notion of self-assembly is very much a kind of a bottom-up kind of process. In terms of Lou, in terms of individual based modeling, that these are agents individually interacting with one another and so on. And it's a great deal of biology, ribosomes, membranes, three-dimensional structure of proteins, virocapsids, etc. kind of thing. That the much of biological organization and what we view as beautiful organization in biology comes about by spontaneous self-assembly. This is an entropy rather than an enthalpic kind of reaction. You're not doing work in the usual kind of sense. And within viruses, most of them is you can view the individual protein capsomeres as trapezoids, and they can go into either these kind of triangular or pentameter kind of associations and self-assemble into a particular kind of structure. And this just shows you a kind of a kinetic pathway of this kind of folding. This one shows frequently, for instance, like with the RNA viruses, that the RNA is attached to the protein, and so there's some beautiful graph theoretic work by Twerk, and she's shown that's kind of a Hamiltonian path in terms of this kind of folding and so on. But in 1982, the Nobel Prize was given to Aaron Klug for his work with Don Caspar and what are known as the Caspar-Klug equations and Caspar-Klug theory, that basically most of the viruses were icosahedral. Instead of a simple 20, most of them were multiples of 60 going on, but all you needed was to have 3 trapezoids to make a pyramid to think about how this might come about. And so many students learn the basic kind of math of crystallography, and down here in the lower right you see a T number of 4, where you have 4 equilateral triangles composing one, and then we have various kinds of classifications of icosahedral kind of viruses obeying the Caspar-Klug kinds of situations. And visualizing it in a diagrammatic way on the left with an electron micrograph on the right in terms of how to see these multiple forms of symmetry in many of these kinds of structure. Well, recently a new kind of printing became available that we could move from 3-dimensional printing so these are 3-dimensional printed viruses, and you know they're static and so on. They of course are beautiful, you know, and one can study the kind of symmetry structures and so on. But it's very difficult for students to understand what's the process by which these things come about. And not only students, but so on. And I said I was very much influenced by Govind Menon at Brown. This is a PNAS paper of his on kind of self-folding. I've taken the approach of self-assembly rather than self-folding, but I'll talk a little bit later about self-folding because I think there's some really important things to be learned looking at self-folding versus self-assembly. But 4-dimensional printing is the 4th dimension is time. So we print 3-dimensional pieces, and then together we use a temporal process to do it. So hopefully the movie will work. So here you've taken a polymeric material, you've put it in water. The hinges are hydrophobic, and so over time at the hinges you tend to bend. So you went from 2 dimensions to 3 dimensions with time. This is Schuyler Tibbets at MIT, who's been one of the pioneers for it. I had trouble downloading this video, but it's here on the one. In this case, you can take a linear one that then folds into the lattice of a cube, but you can go online and see this kind of thing. And again, Govind Menon has done this beautiful kind of thing of looking at kind of various kind of pathways and so on in terms of doing this kind of structure. I've been particularly interested in the problem at the very top. In protein folding, we do what are called HP lattices, where we assign each amino acid as either hydrophobic or hydrophilic, or polar or hydrophobic. And how do we take a linear chain and fold it into a secondary structure or sometimes into a 3-dimensional lattice. As far as I know, there's no good software for taking any given linear protein chain and drawing it out as this kind of thing, and it's been one of the challenges I've tried to work on as a software developer to do, and I have not good enough rules at this point to do that. But Olson at Scripps Institute Oceanography built this structure in another PNAS paper. So here we have a dodecahedron that we simply have our 12 pieces, and unfortunately I've lent this out so many times somebody lost one piece. But you can see that within a short period of time, I was able to assemble a three-dimensional version of a viral capsid. But as fascinated as I was, so break it up hard first, and then I usually recommend a horizontal swirling motion kind of thing for success. But most students can be successful. And in addition to publishing a PNAS article, that they also did a CBE Life Science Education article in terms of students learning from a static 3D model or a 2D model, or doing origami, taking a 2D piece of paper and then folding it into 3D. And I brought some of those along as well, kind of thing. And they also, you know, you can look at videos here where they had this. One of the most outstanding is they did 2 different isomers, and that you could have a yellow and a green version that in the same bottle, and that you would get a completed yellow one and a completed green one. I've not found a video of that, and I ordered the other one, and the company has not been delivering for several years, kind of thing. So I'm going to have to build it, you know, kind of thing. So anyway, that little green bottle that cost about $90 for me to get that one commercial one, and so on. And of course, the first thing I tried to do was duplicate it with my 3D printer and so on, and had a lot of trouble repeating it. And I had 4 undergraduates, 3 from biomedical engineering and 1 from mechanical engineering, working with me, and they almost wanted to give up. And you know, you're trying to use SolidWorks to design the 3D pieces and get the angles all right and so on, and then you're drilling holes and gluing in magnets and all of this kind of stuff. And it wasn't working. And we went through their patent, we went through their article and all this, and the students were about ready to give up. Well, as usual with students, they finally listened to me and did a little bit of mathematics, and so we did what are called Dürr nets and Schlegel diagrams that I'll introduce to you, as well as looking at the Kasper-Klug theory, et cetera. But some stories emerged. So first, a little bit about the icosahedron. It's a wonderful structure in terms of thinking about space and so on, that you're able to get a fairly large volume with a rather minimal kind of surface area in terms of thinking about this. That sometimes within a given bacterial cell, you can have as many 40,000 of these viral capsids in a 1 micron by 1 micron by 2 micron box. That's pretty impressive as a self-assembly at the nanoscale. And so one of the things I've tried to get interest is we have a huge center of nanotechnology, is that this is part of a new material science, and I'm particularly interested in the notion of biomimetic design. But first of all, by even taking the solid 3D one that you have, if you examine it, we have 4 kinds of symmetry. And so if you usually think about 3 protein molecules consisting of 1 triangle, we can have a symmetry of 2 or 3 or 5 or 6 on these kind of things. So it's really kind of amazing that you have so many different kinds of possible symmetries with such a simple kind of structure. But, you know, students are always kind of weird, but in 1528, Dürer, the German artist, many of you may know his self-referencing hands drawing kind of thing, but he published this kind of weird polyhedron, but he also showed the artists how you could do a 2-dimensional origami pattern for it to fold it. Now, do you have it back as a single egg yet? Oh, okay, we're oh, well. What kind of student are you? Okay, and so these Dürer nets are of some interest, and so here's a typical kind of thing for high school in terms of how to design your, you know, polygons to have this kind of polyhedron and putting flaps on it and so on kind of stuff. But the other projection is topological, from the 3D to the 2D in who's next to whom, okay? And so these kind of projections are very nice. They're called Schlegel diagrams. And so one of the ways that I try to introduce them is I build tinker toy kind of models and then take a flashlight and look at the shadows, and you can kind of see a projection that while it warps all of the distances and angles and so on, the topology is reserved. Or as one of my colleagues said, topology is geometry in California, all twisted and bent out of shape, you know, kind of thing. So you know, that one of the things I also became interested in designing these things is the notion of kinetic traps. And show you that very frequently there's intermediate ones where you have to break it up and then reshake it in order to get the final structure. That certain substructures are an impediment to reaching the final structure, okay? So how could we make a kind of a better model? And, you know, setting out these poor students to do this kind of thing. And so one of the first things was designing literally the magnet map of how could these pentagonal pieces reproduce to do an dodecahedron. And so having north and south poles, where do we distribute them and so on. And if you look at it here in the center, fingers are too big, Chris. Why am I? Finally. So can you see that this pentagon in the center of the two poles of this dodecahedron are different than all of the other 10 tiles? And you could have them of two types, one with all north poles and one with all south poles, okay? And so you can make two different dodecahedra depending on whether the nucleating pentagons on the two ends are differentially oriented. Does that make sense? Okay? So you only have two kinds of tiles, that 10 of the pieces are identical and the two poles are different, okay? But what wasn't present in the article was always clear of who is attached to whom in this, and so we frequently had to do these kind of drawings of it. And so the benefit of moving from the Dürr net to the Schlegel diagram is you preserve that kind of thing. And so in terms of trying to build more realistic kinds of models of the viral capsids, we tried various different kind of structures. And so we tried dealing with trapezoids, we tried dealing with equilateral triangles, we tried to deal with these five-sided kinds of things and so on. And one of the problems we had with these structures, and Lou, is that this one we glued together, we could never get to self-assemble. The acute angles in here really turned to be problematic in a shaking kind of environment. And yet there's good X-ray crystallography data that there are intermediates inside the cell of this type of structure. So we haven't totally given up on it, but we think we have to do some different kind of design principles. But as usual, we were talking earlier today, and Lou and Raina and myself, about failure and how these failures really help inform you and what you're doing. And even in producing the dodecahedron, we had some really severe kind of problems. But in taking a pentagon and making it into 5 triangles, one of the things you see is you get some 3-dimensional buckling, okay? You're not, that if you had 60 degrees, obviously you would need 6 triangles to have 360 degrees. But that if you can do it with buckling, you can do it with 5 triangles, okay? And so one of the things then is that pushes you into the 3rd dimension. Instead of printing a planar piece, you now have a 3-dimensional convex surface. And what we didn't realize how important that convex surface was until we got further in the project. And so we started to deal with different kinds of structures and so on, and certain things didn't work well enough that we felt primarily we had some problems with symmetry and so on. and a variety of other kind of failures. So we tried some with, here you can see that we had 6 trapezoids, not just 5, and mixtures of 5s and 6s and so on, and trying some of these kind of structures. Well, finally we had success. Partly, what was the weight of the plastic that we were dealing with? What was the strength of the magnets relative to the weight of the pieces? This notion of pushing it into the 3rd dimension of a complex surface. If you look at the green bottle going around, you'll see some little bubbling. And I, unfortunately, and students initially presumed that it was kind of decoration to make it look like the X-ray kind of photographs that we do. But it turned out to be a very critical kind of piece. So here was the Olsen kind of thing. And here's 8 of the models that we dealt with. We dealt with some others, but they weren't even pretty enough to show, kind of thing, you know, kind of stuff. And then we tested our models with a variety of people in terms of how they were working and so on. But we began dealing with that we had to understand something about having stronger magnets relative to the size. We needed to have kind of symmetrical shapes. They tended to be a little more circular, smaller, and what was in particular was the convexity of the external surface versus the internal surface of the icosahedron or dodecahedron. And a wonderful article in Chemical Science called Ship in a Bottle, Confinement Promoted Self-Assembly, dealing with it in chemical structures. And the confinement effect renders complex self-assembled species robust and persistent under conditions where they do not form in bulk solution, kind of thing. And so I found this video online, and it was truly transformative. And so I then started collecting bottles of different sizes and shapes and so on, that it's truly a ship in a bottle kind of problem. And so in terms of doing it, I would like to share our success. And my students titled it, "You Can't Make It Unless You Shake It," and so they call the model Shake and Make. And so here is an icosahedron self-assembling, okay? That one I'll throw, but the next 2 I will not throw around. But then we experimented with different kind of materials. This is a flat bottom, cylindrical flask, and this one is truly round bottom. And so to study some of the kinetics of this, that literally going from the flat bottom cylinder to a flat bottom sphere to a circular bottom, that the kinetics is that the average time to assemble an icosahedron went down sequentially on these 3. Now, I tried about 11 kinds of bottles and so on, and trying to find Christmas egg ornaments that are Easter egg ornaments or Christmas ornaments that were transparent that you could put things in and so on. These are the kind of things that my wife said, you with your toys and going to craft stores and kiddie stores and toy stores, kind of thing. But actually one of my colleagues who works with kids says, oh, you need to sell this to Toys R Us. So Lou, you can pass these around, but be a little more fragile, kind of thing. So we have successfully done, and we realized that, that in order to do this, that we really needed to understand some more about the dynamics of self-assembly. But we think that having a dynamic model of this kind of process, that we have a better potential for people understanding this kind of entropy kind of driven kind of process. Where do we want to go? Well, we want to build ones that have 60 capsomeres. We want to build some that have the mixture of pentamers and hexamers of the trapezoids. But we also want to do some threaded kind of structures, like attaching a string with the RNA along the protein capsomers, and how is this string set of pieces assembling. So they're tethered together from the beginning, kind of thing. And we're also interested in some structures that violated the Caspar-Klug theory. And so Ron Twarek in England has really done some beautiful work on quasi-crystal models of viral capsids, and also finding some structures that supposedly couldn't be done with Caspar-Klug theory, like T2 and T6 kind of capsimers. And she dealt with two types of tiles, kind of what referred to as kind of kites, and that this is a model of SV40, and this is kind of the Penrose tiling quasi-crystal kind of thing. I think they're absolutely gorgeous, and another problem really worth doing. So here's her solution in terms of doing these two kinds of four-sided structures that self-assemble into it. Now, she does it theoretically. I haven't seen anybody do a 4D printing of this kind of thing, but it's one of the challenges that I'm trying to work on. And I was also interested that some biologists were using things that satisfy the Caspar-Klug things are known as Goldberg Polyhedra, known long before Caspar and Klug, but kind of rediscovering, and that some biologists have used it to study other kinds of structures. And so think about clathrin cages and other kinds of things that occur at the molecular level of polyhedra. But Govind Menon and his students have been doing this wonderful stuff with hydrophobic polymers at the micro scale. So they literally are photographing this at, say, 100 micron kind of resolution under a standard microscope kind of thing. And they've tried to study preferred pathways. And so if we look in the upper right here, for a cube, there are only 11 distinct Dürr nets for origami folding a cube. Here for an icosahedron, and we could also show for a dodecahedron, there's a combinatorial explosion. So a tetrahedron has two solutions. A cube and an octahedron each have 11. The dodecahedron and the icosahedron have 43,000 Dürr nets. 43,000. You go from 11 to 43,000. So here are the number of nets. 2, 11, 11, 43,380, 43,011. If we go to a truncated octahedron, Govind has said about 1 million, and for a viral capsid on the order of 10 to the 30th, okay? So very, very interesting, but even, ooh, ouch. Let's go back and come forward. Okay, but in terms of assembly things, what I wanted to show you why the icosahedron is so much more interesting to me than the dodecahedron, because we have 57 billion versus 18,000 different assembly pathways for the icosahedron. So you can see why I'm intrigued of this as kind of a structure. So others have written about this difficulty of the self-folding, and so I simply want to read this one to finish this first section. Why is it difficult to refold a previously folded sheet of paper? We show that even crease patterns with only one design folding motion inevitably contain an exponential number of distractor folding branches accessible from a bifurcation at the flat state. Consequently, refolding a sheet requires finding the ground state in a glassy energy landscape with an exponential number of other attractors of higher energy, much like in models of protein folding, Levinthal's paradox, and other NP hard satisfiability problems. As in these problems, we find that refolding a sheet requires actuation at multiply carefully chosen creases. We show that seeding successful folding in this way can be understood in terms of subpatterns that fold when cut, folding islands. Very frequently I talk about kind of kinetic impasses and so on. Besides providing guidelines for the placement of active hinges in origami applications, our results point to fundamental limits on the programmability of energy landscapes and sheets. And so this is my team that worked with me, this was, she's a mechanical engineer, and the other three were biomedical engineers, and this was their final poster for their senior project in terms of doing this. And great fun of working with them. So questions on this first talk? And yeah.
So if we bring it to the biology, so how quickly can those things self-assemble? So will that help you to understand the process, like, you know, building this in tubes, etc.? Because I don't even know how quickly they're assembled in vivo, like those kind of structures. Is it milliseconds, seconds? Is that how do we learn?
Remember
the biology part?
That in fast culture, an E. coli can reproduce a new progeny in 20 minutes. We can produce about 40,000 viral capsids in the same period of time. That's pretty amazing. Well, within one cell. But one of the things is, I would argue that probably in terms of multiplicity of infection, the titer that comes out of it, that in terms of natural selection and infectability of new cells, only a very small subfraction of them need to be fully assembled into, say, an infectious form. That we have some preliminary data from colleagues that with X-ray crystallography that there are all kinds of half-assembled, deformed kind of capsids, et cetera. And so some of the kind of kinetic traps. So if I can have one of the icosahedra back, please.
Do you have a preference?
No, any icosahedra. So,
How many rings is that supposed to have, Shelby? Do you know? I mean, I'm looking at it.
It starts with an A.
It starts with a single egg. It's supposed to go back to one single.
Yeah, but the wires walk. I mean, how many wraps are there when you cut it together?
so if we take 5 of the triangles, you can see it makes a very nice pentagon.
Yeah.
And so an icosahedron is 20, so you would think we could take 4 of these, 4 times 5 is 20, but this is a kinetic trap because 4 pentagons will not cover the dodecahedra. By Euler's rule on vertices, edges, and faces, we can't do it with less than 12, okay? And we only have 4. And so if you just form 4 of these, or even you form 1, you have to kind of shake and break it apart because these are not good substructures of it. You can deal with at most 2 of these, okay, in terms of trying to build an icosahedron. So this is what I refer to as a kinetic trap.
Does that happen in biology?
Yeah, and so that's
What happens to
with
those?
some of the people beginning to understand self-assembly, and that's why I showed you the slide on this notion of kinetic traps. That and so one of the things I love, and so as future work is, let's compare self-folding and self-assembly. That since I've got 43,000 different Duryer nets that I can deal with, that what happens if I take a long linear strip? And so if I had more time, I'd take a strip of paper and I would do an origami fold to fold it into a pentagon. And then can I just keep stringing a sheet of paper and keep refolding and have a string of 12 pentagons that then that strip becomes a dodecahedron kind of thing. I can't as easily do it with an icosahedron. But, you know, these are kinds of puzzles that, you know, I sit there and, oh, John's playing with his paper dolls again, you know, kind of thing. It's your colleagues looking at. But I think there's some very deep and fundamental insights into some of these problems. Yeah, Eric.
So you were saying that the shape of the container had a large impact.
Yes.
is there a correspondence to the biological shape where these
Yeah,
are forming? And it's sort of a three-part question. And have you looked at 3D printing containers so that you can make whatever shape container you want? And thirdly, have you looked at the viscosity of the fluid in the container, and does that have an effect?
I think exactly. So remember that you're in an Aristotelian world at this size. So Purcell's famous kind of work, Life at Low Reynolds Number, that in terms of the dynamics of the physics here, is you don't go anywhere unless you're pushed, okay? And diffusion does not work, okay? And so when you're at this scale in a fluid environment, the biophysics is such that you have to think counterintuitively to your macroscopic Newtonian world. And so I think it's really important to think about that you're in a non-Newtonian low Reynolds number world where viscosity, buoyancy, things of momentum are counterintuitive to your intuition. And so that's why it's really important for me to how do we explore thinking in the nano world.
Just curious, like if you double the number of pieces, would they form into two?
So that's what we want to do, is triple the number of pieces. We want to go to 60. Okay? And so one of the things we tried doing initially was building 30 dimers, so 2 triangles together. We've gotten some interesting substructures, and I'm going to try some tricks up my sleeve and see whether I can't do some progress on that. But you know, one of the things, think about what if we had 2 sets of strengths of magnets that I have one set that I form 3 trapezoids into a triangle, and then a weaker set for triangles to self-associate with triangles, that I'm more likely to form 3 trapezoids into a triangle. And so if I can use 2 different set of, think of it as bonds of different strengths, then I might be able to do that. And one would think surely with viral capsids, where you have capsomer proteins, that those inner molecular kinds of strength of those bondings are going to be different. And so all I think I need to do is be just qualitatively slightly different, and I want to try that, so, and in terms of doing it.
Just for some of the postdocs that are here, I've developed this simple thing for middle school students, hands-on activity with, and these are all built with equilateral triangles. And so you can, in talking about what you can build with equilateral triangles, and it's very, I don't know if you've ever done this and build 3-dimensional solids and, you know, and convex, convex solids. And anyway, it's pretty interesting. It turns out, you know, there's certain things, you know, if you have, you can only, the biggest you can build is with 20 triangles, right? And they go up, you know, and you have to have a even number of triangles, and you can't do one with 18 triangles. Have you ever done this, or are you familiar with this? Yeah.
Yeah. So
anyway, I have some activities like that. So if any of the postdocs here want to, I have some other activities like middle school that I've actually never demonstrated. Maybe sometime we should have a session here where Suzanne demonstrates some of her middle school stuff. So
I almost brought along one more set of toys, but they're too big and too heavy for my luggage. But they're called magneformers, and so. That literally I bought an excessive numbers of equilateral and these triangles and squares and pentagons and hexagons. And one of the nice things is have students build convex polytopes and then break them apart and lay them onto the plane so that they have a notion of the 2-dimensional projection onto, say, a Dürr net, and then to think about how you refold that into 3 space. And so it turned out that I do a lot of workshops on STEAM, art with science, technology, engineering, mathematics with high school teachers in interdisciplinary contexts, and they fell in love with these magneformers and making different kinds of structures and so on.
Probably the magnet ones cost a lot more, so I bought the click together ones.
Yeah. Well, magneformers are so. Take one last question, and then I'll try to condense the next one into about 10 minutes.
Just really quickly, how long did your students work on that? How long did it take?
It was supposed to be, September through December. I would say at most I had about 8 weeks of their attention, kind of thing, because of the other requirements of the course for senior design that they were in, kind of thing. So, I was pleased that in a semester we had that kind of progress. But came Thanksgiving, they were totally discouraged and ready to quit. And 2 weeks before final poster presentation to a large audience, they finally had some. But again, it's that we're going to do some little mathematics before we're going to go on, kind of thing. You're not just engineers sitting there with your SolidWorks designing it and running your 3D printing. Let's think conceptually about this problem and what's going on a little bit. So, fair enough? Of any interest? Okay. So, the other part of the nano world that I work with is 3D nano tomography. And so we're able to use X-rays to literally put together, just like you would go and have a CAT scan or an MRI. My wife recently had one of her knee replacements with a 3D scan where they would then 3D print a replica of her knee and less surgical time. Unfortunately, then the hospital changed rules and wouldn't allow, even though the physician wanted to do it. But I think because it had less operating room time and lower billing, that they weren't willing to let him continue to do this kind of thing. So the vagaries of modern medicine. But I've been interested in biological image analysis for a long time. I got very much in love when all of a sudden a tool like NIH Image came available probably 30 years ago, and it very much transformed kind of my life as a microscopist in terms of doing it. And I had been working on a problem with cancer of the tongue. And if you think about it, smokers and betel nut chewers and a variety of other people that various, even kind of food metabolites, is the tongue is a source of a lot of cancers. And many people die. It's just behind melanoma, and it's more common than leukemia kind of thing. And one of the changes for many of you is if you go into the dentist now, most dentists will pull out your tongue and will do an examination for tongue cancer. And so with image analysis, we're interested, can we go beyond what the cellular pathologist does in the lab, and that they can look at a few cells on a few slides, and we're interested how could we have thousands of cells from multiple sections analyzed, and that we might affect the notion of diagnosis and treatment and prognosis kind of thing. And so this has been a long, interesting thing, and particularly in Scandinavia, there was a variety of kind of work using some stuff that's been dear to my heart, of using geometry and topology and spatial statistics. And so you would find cancer slides like this, where you would do a Voronoi diagram, which is a nearest neighbor polygon. You simply, where you have 2 points, and you have the perpendicular bisector between them that you've divided up the area between, and a Delaney triangulation, which is the topological dual. But we also can do various kind of graphs on these things, like we can do a minimal spanning tree and so on. One of the trees that I've been very interested in is called a Ulam tree, after Stan Ulam, and there seems to be very literature on it. But when you look at these kind of structures, very frequently people call them broken, deformed, irregular, asymmetric, non-uniform, imperfect, etc. To me, I mostly call them beautiful. So here's a wet lab simulation of it. You're just dropping ink onto agar, and as it diffuses out, then instead of having circular boundaries, people are quite surprised that you get these kind of lines. I can do it in 3 dimensions, so if I do a face-centered cubic, I didn't bring all my toys along, but if I take one Play-Doh and then I put 8 on the cubic corners and then 6 more on the faces, so I have 1 in the middle, 8 plus 6, as I have 15 balls, I crunch them together, and when you cut it in any orthogonal plane, you tend to get 5- and 6-sided nice straight lines as you push these balls together. Very counterintuitive to people that we start out in this 3-dimensional structure and we get these kinds of things. So it might not just be a 2-dimensional epithelium, but so on. But one of the things that surprises people in teaching them something about image analysis, that just by looking at an image, I say, what's the causal mechanism that's likely to have brought this about? If it's long-term, and so take some spaghetti before you boil it and just drop it, or very frequently I used to use pickup sticks, okay? You can see that statistically most of the time you get 3-sided and 4-sided kind of polygons, and the junctions are X junctions, versus if we go back, right, we can see there, but you can see most of these junctions are trigonal, and in fact the trigonal point between 3 adjacent neighbors is the circumcenter because it's equidistant on the radii between all 3 generator points. And so in a nearest neighbor pattern of points, point interaction rather than line-line interaction, you're going to statistically get a predominance of 5- and 6-sided polygons versus 3 and 4. So this is what surprises many of my biological colleagues when I talk about the power of mathematics, that simply having the model that nearest neighbor interactions of local versus global kind of long-term lines is I can take a piece of pottery with a glazing on it and look at it. Was this mostly thermal effects that are local, or is this long-term cracking, et cetera, kind of stuff? So this goes back to 2 mathematicians, 1908 and 1934, that these Voronoi polygons are simply generated. The red points are the circumcenters, the green points are the generator points, and the black lines are the edges of the Voronoi tessellation. And then we can also do the topological dual. And the reason I stress a lot of the topological dual and a lot of working with topology is that in many cases, if you're dealing with different sizes and so on, you want to be interested in who's next to whom and so on kind of thing. There's also some real pragmatic things. Let's say that you had a distribution of sensors, or you had, say, even just rain gauges set out that if one of them has a leaf covering it or something, or goes out on you, you could just cut those particular ones and recompute the local kind of environment if you're trying to do, say, the area and so on. So having both the topological and the geometric. And so in 1908, Voronoi came up with the Voronoi tessellation, and the 1934, the Delaney thing. One of the nice histories of the Voronoi tessellation is the classic case in epidemiology of John Snow's pump in cholera, is that unfortunately the popular book on the ghost map does not actually show the Voronoi tessellation. So of course I had to do it, you know, kind of thing. So I did a numeracy article on that kind of thing. But one of the things for me, and so even if I don't get through much of the rest of the talk, one of my primary rules is in trying to find mathematics that are appropriate for my biological colleagues. My rule of 4 is that in distinction from the physicist that's interested in precision, prediction, and accuracy, that for biology, my model, if it works across a broad biological diversity, across phylogeny, across various spatial dimensions, temporal dimensions, and across different kind of biological causes. So it might be diffusion, it might be growth, it might be a fair division of, you know, this fish with that fish, building a boundary halfway between them, et cetera, that these 4 rules seem to apply to all kinds of math modeling that I do, and I've found real richness in dealing with these kind of cases. So here's a case of time that going from femtoseconds of some molecular kind of structures to looking at a tree canopy in New Zealand and in Thailand and Malaysia, where it takes maybe 300 years to have a mature tree canopy. And here the trees, they might be growing out this way, but you have polygonal boundaries when you look up at the canopy gaps. And across phylogeny, and some of you old enough who remember the famous Powers of 10 movie from Grant Park in Chicago, with physicists going from the atomic out to interstellar space, that in biology we can match this with 24 orders of magnitude just as well, kind of stuff. And so the spatial temporal dimensions are there. And the kind of biological processes that can divide these are similar. So with 3 of my colleagues in Thailand, we built this software that we published in Bioinformatics in 2012. A biologist can simply import an image, and then you just click on, say, cell nuclei or seeds on to the surface of a strawberry or individual trees in a forest or whatever. And then we have up here in the software that you can do the geometric and topological kinds of things, variety of different kind of graph theoretic and computational things, some spatial statistics. And here you can see that here we're tending to get 5, 6, and 7-sided polygons. So we have some other kind of statistics. I particularly usually are interested in 3rd and 4th moments of kurtosis and scutosis in terms of looking at this. You can also just use it for a mathematical problem generator because you can add random points and begin to look at it. And down here in the corner is what's called a Pitaway violation, that in the Delaney triangulation, if we connect these, triangulation would only go across one boundary. But if you have a situation where you go across multiple boundaries, that's a Pitaway violation kind of situation. And so very frequently we're not seeing it here, but if the center of this cell is here and here, we might go across 3 different boundaries, even though they save one here. So, and it turns out that Ulam trees and Pitaway triangulation violations are 2 things not sufficiently in the mathematical literature that I've been interested in exploring. But with our software, these are 8 basic things of image analysis that I tend to look at. And so is there a relationship? And biologists observe some of these first, and some of them are more famous coming from the math audience. But is there a relationship between perimeter and the number of sides, area and the number of sides? That if you tend to be a big one, do you tend to have a bunch of small ones around? So this is an important theorem, the Weir-Phelan kind of foam and so on. But these are kinds of things I've been very interested in looking at these patterns for a long time. And so was having trouble with the cancer problem, so where do you turn? Embryo Genesis, that every mother knows that this is a foreign body growing rapidly as a group of uncontrolled cells inside their body over a short period of time. And so this tumor called a baby, is something that's a very similar kind of process of going on. And so I turned to Drosophila eggs, and that you have to understand, the size of a fruit fly is small enough, but you're literally trying to examine an egg, and we literally have to catch it almost as it comes out of the ovipositor of the mother kind of thing. But a wonderful image was, oh, ouch, let's hope this is loads, from a book on nano nature, that the wrinkled texture, hairy orifice contrasts starkly with a smooth, sleek lozenge emerging from within, the filigree tracing a ghostly mesh of hexagonal lines across the glassy surface adds to the disparity of the image. Strange though it may appear at first sight, here's caught an act rarely observed, yet so common and mundane, the laying of an egg. Well, unfortunately, that violates Euler's principle. You cannot cover any kind of complex polyhedron, with all hexagons, and if you look at closely, here are a few pentagons there on the structure. But this author, the artist, was not alone. Here was a famous article in Animal Behavior that called the hexagonal cells of these mouth-breathing fish in terms of whipping up the muddy, sandy bottom between their nests. And, I simply highlighted 5 pentagons amongst them, kind of thing. So But more recently, an article in Computational Biology and looking at these, the typical Harvard thing, and unfortunately, I want to show the emperor has no clothes, but I can show some things wrong with their model, but I haven't had a good alternative model, but I now have some Belgium colleagues who have literally done some microscopy of looking at actin filaments inside of cells and found a difference in the way of division of a fruit fly egg and a flower beetle egg. And so we've been looking at that. So here's doing a Voronoi tessellation on 5 successive divisions of a fruit fly egg and looking at their distribution over time kind of thing in terms of what's going on in this thing, and then testing it with various ones of our parameters and so on. And so here we're comparing, fruit fly with flower beetles. The problem with both of these, and also then I tried plant Arabidopsis thaliana, kind of the model plant for developmental biology. And I really, even with a laser confocal microscope, couldn't get good 3D. I was worried that I had these curvilinear problems at the edges of these, ovoid-like structures, and what things were artifacts of that. I was dealing with a 2-dimensional picture of a 3-dimensional thing. So I even tried some scanning electron microscopy where I could stage it. I was trying to do some z-axis integration on confocal microscopy, but we began looking at a variety of these things. So here's implementing these Ulam trees and looking at these topological charge and so on. But another problem that you have is in 3D, a problem of parallaxis in terms of getting accurate measurements of you projected onto a plane, and does that really represent the 3-dimensional distance? And a problem of Delaney triangulation is what's called flipping, in terms of when you have 4 points, that just moving them a little bit one way or another, you go from a vertical to like this horizontal one. And so in some cases, the topology is changing because of the geometry. And so we suffer from that. So I went back to a beautiful old friend, and these are the radial area. For most of us who are interested in art and biology, Ernst Haeckel's drawings from the 19th century and his book Art Forms in Nature just stimulated many of us, and I'm finishing an article that's a anniversary homage to Haeckel right now that should appear soon, but kind of thing. But so you get some sense. There's thousands of species, and these are basically amoeba with glass houses. And they're all marine, and usually you're picking them up in sediment. I had been buying it from Carolina, but they were embedded in plastic on fixed slides, and trying to remove them from them and look at them under my scanning electron microscope is even difficult and so on. But finally, a retired colleague in Wyoming had some washed specimens that were from the Glamere Challenger of deep ocean dredging kind of thing, and shared with them, and we could wash them. We actually would put them on a sticky note and send them in an envelope over to Belgium to our colleague for 3D Nano Tomography. And so this gives you a sense of some of the beautiful work of Ernst Haeckel, a variety of books. Hundreds and thousands of his images are available easily on the web and so on. And it just influenced many of us for a long period of time. So, Ritterbusch, back in one of my favorite art science journals, Leonardo, critiqued Haeckel for the kind of artistic license. And so using a software, I showed one of his images. So here, this particular one in the classic case was Alonia hexagonia, covered with all hexagons. Well, you can't occur mathematically. And so Martin Gardner reported the classic story, showed that God was better than the mathematician, and said no, it just showed the biologist wasn't as good at recording what the structure actually was, kind of thing. And yet when I analyzed a different one of his structures, this is much more the typical kind of Voronoi distribution that you can see. Predominance of 5 and 6-sided, some 7, but no 3s and, only a few 4s and so on kind of stuff. And if you look at the famous Glashka glass of the 19th century influenced by Hickel, didn't try to do pentagons or hexagons. It's all a triangular mesh in terms of doing it. And one of the beautiful things at Delaware, we have these art conservationists. And so literally they exposed me to literature on the preservation of this 19th century glass on show at British Museum kind of thing. But it also influenced architects, and one of the most famous one was René Binet in France, which led to this entrance to the 1900 exposition kind of thing, influenced by a radial area. And if you look at it, again, it's not hexagons and pentagons, it's again using it. And so one of my themes here is if you actually build a three-dimensional structure, whether it's the Blaskas in glass or here in concrete or in metal kind of thing, you can't violate, Euler's rules kind of thing. You have to really do it. But it had a huge impact on architecture and so on, and this notion of biomimetic and so on. But this shows you some of the best of what we can do with light microscopy, and these are most of what's available online and people trying to do taxonomy and so on. But Dennis van Loo in Belgium had built this X-radia machine, so with X-rays that we could do nano tomography, three-dimensional, and what we're pleased with is with his company, he's actually used our images for doing it. Give you some kind of sense that to be down here in the nanometer kind of a range, that we're literally dealing with X-ray kind of visualization. But the beautiful thing by having a glass test is that it's hard enough that we can get these high resolution images. So a couple of years ago, we got the cover of Microscopy Today. And so I brought along that, look at that, that's a Russian doll. There are 3 tests, a test inside the test inside the test kind of thing. And Lou, that think of it that I've studied these things for 35 years under a microscope, and now to hold it in my hand and twist it and turn around, the power of three-dimensional printing is enormous. But I want to talk about it as a change agent even more. We can do, so we use a software called Embryo. We can make all of these, avoid the problems of parallaxis. We can do actual three-dimensional measurements in terms of these kind of specimens and so on. Oops, get some sense of scaling here in terms of that having a radial area inside the O of a euro coin and kind of thing to blow it up, that we're talking about 250 million times in terms of doing it. When we get the stuff from Dennis in Belgium, and sorry going on, and I can shut up at any time, we actually get a complex like this. We have a hard time kind of sorting them out and washing them and sending them, and so we have to literally dissect these out to take individual ones, but then we can get this kind of thing that's pseudo-colored for 3D printing. And then with the software, and one of the things you can see that really the polygonal estimation of these is pretty good. And and then we can do a fly-through as well. So you're literally able to get down and look around and so on, kind of these kinds of stuff. And then we can do a medial axle transform to get the polygonal mesh. And from that, we can do measurements. I also take it in software, look at these kind of distributions. We can also do digital dissection. And so we can literally, if we have this Russian doll of 3 different tests, we can look at them individually with this kind of thing. And so these are 8 that we're able to three-dimensional print. It turned out we had some foraminifera in our example, so we also have 2 foraminifera. And the change agent is now that we can, not everybody has access to $1 million machine, but we can give you the 3D coordinates. You can print them in 3D. You can put it into your 3D software. But even the software takes about a $25,000 micro, and it's about $8,000 minimum for a package kind of thing. But I'm hoping more open source tools and more powerful computers, people coming, is you'll have access to it. But even so, that you can simply send a Shapeways and get this. And so my colleague did pseudo-coloring, and this is the November 2018 Micropaleontology Society calendar kind of thing, and so on. But all of our files, we only have one file up successfully so far, is on Morphosource. This is kind of like iDigBio. And so that both the original three-dimensional voxel files and print files will occur, so that anybody, any place, could then either reanalyze them mathematically the way they want, or they could 3D print them for themselves kind of thing. And I think it's a real change agent. My close colleague that I work with in physical anthropology, she's president of the American Society of Physical Anthropologists, is with human skeletal remains, only a few people had access to the original. But now anybody, any place could have access to making authentic scale size replicas of these things and making all the measurements you want and so on kind of thing. So one of the things I'll end with simply here is that I'm arguing that because these things are many different sizes and so on, more attention needs to be paid to topology rather than geometry. And that vertices, edges, and faces are more robust kinds of features that are less sensitive to these issues of scale and issues of distances of angles and perimeters, volumes, et cetera, kind of stuff. And so here's a Schlegel diagram done by Japanese people, and I didn't become aware of their work till very recently, but beautifully that they could actually do a full Schlegel diagram of one of these. And you have to stitch these two together, but this is our Schlegel diagram of one of our full, larger radiolarians. But I'm interested in, Ben was a friend while he was alive of David Raup, who developed this notion of theoretical morphology of seashells, and with my colleague Joe Ellis Monahan at St. Michael's College, she's published Mathematica files for printing all of these, that the 4 different colors are of the 4 different kinds of brachiopods, bivalves, gastropods, and cephalopods. And with a 3D printer, what I've been interested in, that the equations for making all 4 of these huge taxa are similar, but the 3-dimensional volume is mostly empty. And why haven't those things? So I can print structures from the formulas for outside that space. And what about them that it's unlikely that evolution has explored that morphospace? So I'm very interested in this kind of notion of morphospace. And students in my undergraduate evolution case using the stats package JMP and our package called BIRD on measurements on Galapagos finches were able to do Voronoi Polyhedra tessellations around the 13 species in terms of a morphospace of the Galapagos finches kind of thing. And my final thing that I'll leave with you is when we got the cover of Microscopy today, the artist Bathsheba Grossman printed in glass the radiolaria from our 3D files. And so, Lou, may I replace Jitterbug 20 years later with a new gift? So if you'll walk that around carefully. So thank you very much for having me. And I work with another artist. She's a sculptor, and she's in love with this. And she had a gallery at the Philadelphia airport about 50 feet long, and recently at the Philadelphia fine arts thing of her radiolaria. But it's primarily she talks from the heart. She's very much influenced by Jacques Cousteau, that how can we get people to not just save the whale, but to save these miniature creatures of biodiversity so important to our planet. And she shows with love and beauty and so on. And so I end a quote with another artist that every piece is an experiment and teaches us something about the fundamental nature of form, space, material, and process. In nature, these are one. We are heading in this direction, one small step at a time. Thank you for letting me have one small step. Thank you.
I remember at one point we had the songwriters who were in residence, and I kept saying we need an artist in residence. What happens now?
Well, in 1985-86, I was a Fulbright scholar in Thailand, and I was doing some multivariate statistics for a chemist who was working with Celadon pottery. And some of the clays, after 500 years, were less available for the potters. And he had two potters in his lab. He had a Japanese physicist postdoc on the thermodynamics of the kind of healing of the ceramics in the firing and so on. And I was trying to get him to do some better combinatorial design of experiments so that he wasn't just kind of changing one variable at random after another kind of thing. But having two artists, I came back, and since 1985, I have had artists working in my lab. And generally, I have kind of two math computer science students, engineering, two bio, and one artist. And in a 10-week kind of summer program, it usually takes them about 5 to 6 weeks to discover that they need one another to talk about it. And the artist, you know, might be using Photoshop instead of an IH image or whatever, but we work together in understanding texture and form and a different language and hue and nuance and so on. And so I leave you with a story with one of my art students. She said, John, you know, if this science gig doesn't work out for you, maybe I can get you a job in the art gallery hanging things or something like that, okay?
Thank you so much, John. Really appreciate it.
Sorry to go on, but you seemed like a captive audience. You didn't all walk out, so.
How did they make the one you were passing that cube?
You can simply write to Bathsheba at her thing, and she sells those. So it's laser ablation of the crystal. It has to be very clear crystal. And so the Nobel Prize today of laser ablation, now with a high focused laser, just like they can do laser surgery on your eye, she can take our XYZ coordinates from our file and pinpoint those and laser incinerate it and create that kind of structure. And so here I am 35 years later holding a glass representation. And what's wonderful to me, as much as I love the 3D, here to see the glass inside the glass inside the glass, it's 4 layers of glass. I have the glass cube, and then I have a 3 layers of glass test. And I can see things by twisting this around, and I usually have a light under it illuminating. So take your cell phone, put your flashlight under it, Lou, and you know, that it just truly kind of wonderful in terms of it. The only thing you have to warn you is I get called out every time in the airport when I carry these things through. I don't check it through. And this time coming, what was fun is when I left Dulles, the guy was so intrigued that he brought another one. He says, and he was very intrigued with the 4-dimensional printing and shook up, you know, several different ones and wouldn't let me leave until he played with them and brought this other woman over, you know, to deal with it. But isn't that kind of fun, Lou? So, you know, in terms of math bio, maybe John doesn't do serious mathematics, but I have fun and I work with people I like, and I get to deal with beautiful things. So thank you all very much.